or by injecting constructs or RNA. In this case, at least in these two cases, we are injecting these RNAs and then also the DNA for this construct, which will lead to the expression of RFP under the 807 promoter in retinal ganglion cells mainly. I will try to keep this way of showing the construct we are using at each point, so you don't get so confused. So here in green, we see apical markers. One of them is PAR3, which is one of these proteins here. We can see how retinal ganglion cells, when they start to differentiate, they eventually retract an apical process, and they bring with them a little bit of PAR3 at the tip of the apical process, and then after that they grow an axon. They usually do that. This is very, very stereotyped behavior, and we can see a similar thing when we look at the primary cilium, and this is labeled with RL13B. This is unpronounceable for me, where we see the retraction of an apical process, and then also there is a primary cilium there. Well, we will have to see the whole movie again probably to see that. Sorry. I think I have to remove this. Sorry about that. There. So we will see that the retraction there, and then we see here the cilium, which eventually will remain for some time close to the apical side of that cell where it differentiates. So this is something we've been doing, we've been analyzing and looking at. Then one thing we wonder, of course, from early stages was what happens if we perturb the apicobasal polarity of the neuroepithelium, and so this is something we did using one of these mutants, which is actually affecting one of these proteins important in the apical complexes. This is PALS1. The NOC mutant is affecting PALS1, and we see how here we get some cells, retinal ganglion cells, which differentiate instead of in the basal side of the retina, they differentiate on the apical side, and these apical neurons, they are exactly turned upside down, so they retract a basal process and they extend an axon on the apical side. This was a very nice surprise. So cells are essentially differentiating normally, only that upside down. So we have reverted the polarity. Eventually, we found out that these cells were doing this when they were in contact with laminin that was from the RPE, because the RPE is disrupted as well. And that led to another work by Owen Randett in the lab, who found out that actually laminin is very important for the polarization of these cells, and this is probably what was causing here an inverted polarization of these neurons in these ectopic neurons here. Something else we looked at was the eventual role of the slit proteins, of some of the slit proteins, and in this case I'm showing you here a morpholino, a treatment of an embryo for slit 1B. In the zebrafish, and we are looking here again at confocal images, and again always using this same construct, and we see how when we disrupt a slit 1B expression, what we get is neurons that eventually they make an axon, they differentiate quite well, except that they have a hard time retracting the apical process. Eventually they retract it, but they take very long, and even they make an axon usually before retracting the apical process. So we were perturbing this process, and later Grace Wong in the lab also followed this work and found out that slit 1B was actually the role of slit 1B would be to block encadherin binding at the apical site, so allowing for the detachment and retraction of the apical process. Later with Paola, and this is something actually didn't come out in the paper we published with her. This was her PhD work, and I'm showing just a little part because it's probably relevant for what I'm going to show you later. Trying to disrupt cilia, primary cilia in embryos, and there are a set of genes we know that are important for ciliary transport, and they are essential for cilia maintenance and formation. So if we block them, in this case with morpholinos, or also happens the same in the mutants, you get this particular phenotype of the embryos, which we can usually call ciliary phenotype, which comes together with many other things, many alterations in different tissues, which are related to the lack or the reduction of the size of the primary cilia, which are very important in cell signaling, right? So we see here normal retinal ganglion cell, which is differentiating and retracting in an apical process, eventually growing an axon on the basal side and doing this nuclear translocation towards the basal side of the retina. When we disrupted on Paola, actually disrupted here AFT88 and ELIPSA using morpholinos, many times she found that the cells were taking usually a bit longer to retract their apical processes, and also they were moving sometimes up and down or taking time to translocate basally. This was a relatively subtle phenotype, which can be seen here quantified. So cells were kind of moving a bit more or eventually staying longer at apical positions, but eventually they were also accommodating at the basal side. This is something we actually didn't follow up much, but might be relevant to what I'm going to tell you later. Okay, so this is a kind of summary made a few years ago of some of these findings together with others from other people, showing some of the signals which are important at different stages of the early differentiation of retinal ganglion cells. I'm not going to go much into this, but just to show you that this is something we have been analyzing and we are still analyzing actually at the lab. But I'm not going to tell you much, as you will see, about retinal ganglion cells, because now we have also switched a little bit our interest, or not switched, but actually enhanced, let's say, amplified our interest towards the other cell type, which is very, very particular at the neural retina in vertebrates, which is the photoreceptor. And photoreceptors are very funny actually, because they are neurons in a way. So they have an axon, they make synapses here at the outer plexiform layer, sorry. But they also they keep an epithelial-like polarity and an epithelial-like shape. So they are still bound to other cells here at the apical side of the retina, what's the outer limiting membrane by adherence junctions, very tightly to glial cells and to other photoreceptors. And they have this apical side, which is very, very well developed, and also part of it is a cilium. Actually, it's a primary cilium. The outer segment of the photoreceptor is a primary cilium, which is modified. So these cells have at the same time sort of epithelial polarity on the apical side and a neuronal type of polarity or shape or behavior in the basal side. So that was very interesting for us. So we decided to start to look at that. And I'm going to show you a little bit about this story, which has been developed in the last few years. So one question was, how is this transition between neuroepithelial cells and photoreceptors in the retina? This is something just to show you very quickly how one thing we can see is how the outer nuclear layer is developing at early stages in retinal differentiation from something which is quite disorganized and where we see cells with basal processes eventually to a very well organized sort of epithelium, which is on top of the retina. This we can also follow by electron microscopy, and we see here the cell very early with a basal process, and then we see a more or less disorganized, this is 36 hours, at 48 hours we see a more or less disorganized outer nuclear layer, and then a very well organized outer nuclear layer at 60 hours. Still, something very interesting is that you can see here mitotic figures here, and also if you look, well, here we don't see it, but we still should have dividing cells. These cells are still progenitors. They are not differentiated photoreceptors, and that's a very interesting thing. So what we will analyze here along this the presentation is not post-mitotic neurons like the case for retinal ganglion cells, but they are actually pre-mitotic progenitors, which are already committed to be photoreceptors, but they are still eventually proliferating. Usually they will suffer two cell divisions between the moment they go to the apical side until they eventually start to differentiate. So we were interested in the very, very early stages, and we used this promoter and this construct. Actually, this was originally given to us by Rachel Wong, which has a very early expression, and we can follow, in this case, an isolated photoreceptor, which is progenitor, which has retracted its basal process, which we see here. It's very difficult to see the basal process, it's very thin at early stages, but we see that there is a detachment, there is a retraction, very quick retraction, and eventually a translocation of the nucleus of the cell towards the outer nuclear layer. And that happens in a relatively short time. And the cell once here, in this movie, we could see the cell divided once. We see two cells now because there was a cell division a very, very short time after it reached the outer nuclear layer. So this is more or less how they behave in general, these photoreceptors at early stages. One thing we found that was interesting and was also seen in this movie was that the cells have some processes on the apical side mostly. So they are very active on the cell cortex, and particularly they have these very funny and long processes which we called tangential processes, which are on top actually of the apical side, and they are I will show you at the subretinal space. They are quite long, usually like 1.5 cell diameters, so they can go and reach other cells eventually. That's what we think they are doing. And they are quite abundant already at 48 hours. They start to grow in number from 36 when the cells start to differentiate until later. And we can see there are quite a lot here at 48 hours at the margin of the zone of differentiation of photoreceptors. They usually have more or less the same average length along all this time, around 3 microns. This is what they see on average, but they are quite variable at these later stages. We could see them also by electron microscopy, and I can show you here how they are. They can be quite long, and they are very thin. And they are here at the subretinal space, here and here, at 40 and 48 hours post-fertilization. And something which is very interesting when we see them, and this is quite recent, made by Marija Kojic and Gonzalo Aparicio, where we see by scanning electron microscopy the surface, the apical surface of the retinal neuroepithelium. And this is at 38 hours. We see a few processes, tangential processes over the cells, but then when we look at 48 hours, it's almost completely packed with processes. It's very difficult to see the apical surfaces of the cells because they are completely covered with these apical processes. We don't know what they are, so that's the whole story by now. But they are very dynamic and very interesting, so we are trying to look at them now. So the other question we had was if cilia are important at some point for early differentiation of retinal ganglion, of photoreceptors. And it's important to remind that the outer segment is a cilium, so of course cilia are important for the differentiation of photoreceptors at later stages, but we were interested at earlier stages. And we can see here that 38 hours post-fertilization, this is a very nice image made by Marija, where we will look here at the anterior ventral part of the retina, where the photoreceptors should be starting to differentiate. And we can see here primary cilia, very characteristic primary cilia, suggesting that the cilia eventually exist and they are present in photoreceptors. And eventually Marija looked at that and she found that at all stages where we could see CRX expression, there were cilia. She could also look at them by electron microscopy. They were a bit particular, a bit different to the cilia found in neuroepithelial cells. For example, they usually didn't show a ciliary pocket. We can see how at early stages, like 40 or 48 hours post-fertilization, they are relatively long and abundant, and later they start to become very scarce and very short, eventually to grow again from 72 hours post-fertilization. And this, what we see here, the cilia, which are really in every cell, is the beginning of the differentiation of the outer segment. So we see a decline of the number and the length of the cilia in photoreceptors at early stages until 48 hours, and then again growth to form the outer segment. So are cilia important for the differentiation of the cells? And essentially, not much happens when we block cilia, when we disrupt cilia, again, by using morpholinears to ELIPSA and IFT88. But there is something that happens that we could see, which is that the basal processes, which should retract and remain retracted to eventually have an outer nuclear layer, tend to stay for longer. And we can see here how they are usually longer at every stage, and they stay long for a long time compared to what happens in the normal case where the control, where we see a shortening of this process. I'm finishing, Mike, sorry. So just to finalize, to end, to show you some things related to these polarity proteins, and particularly I will talk to you about encadering, which is ADH2, and just this is the slide just to show you that I'm going to show you results from Morpholino studies, but we have done the same thing with CRISPR mutants, and essentially the phenotype is exactly the same. So one funny thing, one nice thing we see here, and that's already known, is that photoreceptors form rosettes here in the cells. By analyzing these rosettes, we find that here we have the cells very well polarized, and we also analyze the shape and everything, indicating that they are equivalent to the outer nuclear layer. And also we were interested to see how the cells reached there, and if they had processes. And indeed, they do have processes which are very similar in length to the ones we have in the control, in the normal situation. And this is when we use Morpholino to encadering. And they also will migrate, they will move. And this case is a real migration. It's not really a nuclear translocation. And they are very, very active at the surface and with a long processes forming before forming eventually a rosette, which will be formed here by the cells. So this was quantified. I'm not going to worry with this, but essentially the cells do relatively normal behavior, except that they take a lot longer to reach a rosette as compared to the time they take to reach an outer nuclear layer, where they only have to translocate the nuclei. So this is more or less a summary of what I have shown you. We still are trying to look at the tangential processes to understand them better. But this process, this part is the one that is interesting for us, which is quite long in time, actually, several hours that cells are not really differentiating, and they stay there proliferating and accommodated at the outer nuclear layer. So this is just to show you the people who made the work. So this is the group now, and these two made most of the work, which is recently been accepted for the International Journal of Developmental Biology. So it's in manuscript form, you can get it. Magela and Gonzalo, and also show you some work by Paola. So thank you. Sorry if I exceeded my time too much.
Thank you, Flavio. That was absolutely fantastic. And certainly some of that electron microscopy is just stunning work. So I'll hand you over to Julian, I think, who's got some questions, I think, from the chat function, Julian.
Thank you, Mike, and thank you, Flavio. That was really, really remarkable insight from the Morpholino studies. And at this stage, I'd like to encourage everyone to contribute questions if you have through the chat box, or if you can raise your hand through your Zoom console and ask your questions now. Flavio, I did have one that I posted from some previous slides where you had looked at data from IFT88 Morpholino treatment with regards to neuroepithelial cell movement. And I posted my question, and I think it might be inelegant, but particularly the distribution in movements, which is astereotypical, and so there is a broad distribution of different kinds of movements. Is that an indication of a mechanical effect of Morpholino treatment for the particular genes, IFT genes, or is it that stem cells are asynchronized, and so the effect on movement is secondary because the neural stem cells are producing a different progeny in an asynchronized fashion? So do we know much about the function of the gene being disrupted to see this in phenotype?
Yeah, well, let me see if I understand your question. We did see, and this is something that was published before from this Paola's paper 4 years ago, actually, when she finished her PhD. And we did find an effect of cilia disruption on cell type specification. In the retina, particularly, we saw a reduced number, very remarked reduced number of retinal ganglion cells with and concomitant apparent increase in photoreceptors. So the retina was forming okay. It has had all the layers and everything, but there were less retinal ganglion cells, and apparently the balance was towards getting a bit more photoreceptors. We didn't really quantify well the photoreceptors, but that's what it looked like. I can, at some other point, I can show you that. Still, of course, if what happens with cells when we affect them has to do with secondary effect of this change in identity, or if it is more related to something affected at the level of the cytoskeleton or something, we still don't know.
Okay, thank you, Flavio.
Thank you.
I had the video background from a video in which the audience was applauding by snapping their fingers, and I think people might have recognized that reference from a more recent movie. But distractions aside, do we have any other questions for Flavio with regards to this work? I saw that you got some constructs from Rachel Wong's
Yes.
laboratory, and I understand that the zebrafish developmental biology community is very tight knit and collegial, and it's a wonderful forum to exchange ideas as well as reagents. So it's really really fantastic to see. That's really ZFin in action, isn't it? It's terrific. Yeah.
That's right. That's right.
If there are any other pressing questions, I encourage everyone in the audience to type them and send them over, and we can convey them to Flavio, if not now, then immediately after the presentation. So thank you very much. Thank you, Flavio. And Mike, I pass it on to you.
Perfect. Thank you again, Flavio. That was wonderful.
I will stop sharing.
So moving on, our next speaker today is Dr. Marija Kojic. There we go. Hello, Marija. Lovely to see you. So I've got Marija's bio here. She's well-travelled, which is lovely to see. So she did her master's degree with a major in genetics at the University of Belgrade in Serbia, and then went on to Germany to look at actin cytoskeletal dynamics during epithelial morphogenesis. And then in 2012 saw the light and came to Australia and did her PhD at the IMB at the University of Queensland with Professor Brandon Wainwright. I was very fortunate enough to be on Marija's confirmation committee, so I got to see her process and her evolution, and it was one of those occasions where at the end of it you go, wow, what an amazing process that was, to see how Marija developed over the course of her PhD to end up with a project, and which is now continued in her postdoc in Brandon's lab, which is generating amazing data and coming out in some beautiful publications. So I'm going to hand over, stop talking, Marija can show you some of this spectacular data that she's been generating on the Elongator mutants. So Marija, over to you. Have fun.
Thank you. Let me just start sharing the screen. Wow, thanks so much, Mike, for those great words. And I was very fortunate to have you in my, doing my PhD in my committee and now as a collaborator. Thank you everyone today, and I'm looking forward to sharing my story with you. So I'll tell you more about the elongator complex. This complex is a translational regulator, and it's very important for neurodevelopment. Thus, mutations in its subunits lead to a wide variety of neurological diseases. So let me tell you more about the complex itself. This complex is highly conserved in eukaryotes. It is a dodecamer. Sorry, I'm just gonna actually use that pointer that I saw from Flavio. I like it. There you go, looks nice. So this is a dodecamer, as it contains 2 copies of each of its 6 subunits, ELP1 to ELP6. The subunits are organized in 2 subcomplexes, ELP1, 2, 3, which is a trimer, and there are 2 of these in the holocomplex. And this is a catalytic subcomplex, ELP1, 2, 3, and there is accessory ELP4, 5, 6 subcomplex, which forms a hexameric ring. Now, various loss of function studies have shown that each of these subunits is crucially important for the structure, stability, and function of the complex. The complex was initially discovered as a transcriptional regulator, as its catalytic subunit ELP3 is a histone acetyltransferase and is capable of acetylating all core histones in vitro. But then in the last decade, there are various cellular activities assigned to this complex, and these include exocytosis, tubulin acetylation, cytoskeleton organization, but it has also been linked to various cell signaling pathways. Now, there is a growing experimental evidence suggesting that all these cellular activities that have been assigned to the complex are just downstream effects of its main role in translation. Now, how the elongator regulates translation. This is being done via tRNA modification. So the hexameric ELP456 subcomplex binds and releases tRNAs in an ATPase dependent manner, brings it to the ELP123 subcomplex, and then the ELP3 subunit adds carboxyl methyl moieties to uridines positioned in the 34 position within tRNAs, and that's an anticodon loop. So this particular position 34 is also called wobble position of the uridines. And if these modifications are not present at wobble uridines, then this affects translation of codons that are specifically in A and G ending codons, predominantly A ending codons in mammalian cells. Several studies have shown that loss of these modifications, which would of course affect these codons that are A and G ending codons, results in ribosome pausing, and that further triggers protein misfolding response and aggregation. So various mutations in the elongator subunits have been linked to a wide spectrum of neurological diseases. First, these were mutations in the ELP1 subunits, first discovered, that lead to familial dysautonomia, which is a motor neuron disease. Then ELP3 mutations have been associated with ALS, amyotrophic lateral sclerosis. Then ELP4 mutations have been linked to epilepsy, and mutations in the ELP2 gene have been found in patients with intellectual disability. In my PhD, I worked on the ELP6 mutations that we discovered in a mouse after a mutagenesis screen, so it was reverse genetics. We found these ELP6 mutations to lead to a phenotype very similar to cerebellar ataxia. Cerebellar ataxia is characterized by severe motor impairments, loss of gait coordination, and balance. Then after that, so this study was done in mice, but then after that, in collaboration with many clinicians, we identified mutation in the same gene, ELP6, but also ELP4 and ELP2 genes in patients that had not only cerebellar ataxia, but also intellectual disability, autism, and epilepsy. So as you can see, there's so many mutations in elongated subunits leading to a wide variety, leading to various neurological diseases. But interestingly, with the exception of familial dysautonomia, which is peripheral nervous system being affected, none of the mutations affecting central nervous system have been actually studied. So we don't know how these pathologies arise. These are mainly just studies identifying the mutations, but like GWAS studies, but we don't know anything about the pathologies. So this is why we used in our lab CRISPR-Cas9 method to introduce and model these mutations in mice. And then we used behavioral testing, transcriptomic, proteomic, and histological analysis to understand, comprehensively understand the neuropathology resulting from elongator deficiencies. So as I mentioned, first we studied the ELP6 mutations. So this was reverse genetics. We found them in so-called wobbly mice because of their ataxic phenotype. These mutations are missense and homozygous. So all elongated mutations leading to neurological diseases are homozygous mutations. What we found is that the wobbly or ELP6 mutations lead to a progressive ataxic phenotype by using some hind limb clasping, ledge, which is testing for balance, and open field tests. We saw that this phenotype progresses by 4 months of age. And when we histologically analyzed the brain of these mice, we found a massive Purkinje degeneration in their cerebella. So these are principal neurons, Purkinje neurons, principal neurons of the cerebellum. And between 2 and 4 months of age, the cerebellum of wobbly mice becomes fully depleted of these neurons, which is quantified here. Then we used genetic complementation study where we introduced a wild type ELP6 in a PCP2 mini gene. So this is a mini gene that's specifically being expressed in Purkinje neurons, and we managed to fully rescue the phenotype. We also use CRISPR-Cas9 to generate an ELP6 knockout mouse. Now, knockout of any of elongated subunits is embryonically lethal, but we use the heterozygous knockout mouse and cross to the wobbly mouse to get this compound heterozygous mouse that fully recapitulated the phenotype of the wobbly mice. So this way we confirm that indeed ELP6 mutations are responsible for the ataxic phenotype. Then we were the first ones to study elongated subunits in vitro, mammalian elongated subunits, because all the knowledge we have so far on biochemistry of the elongated complex comes from the studies in yeast. So in a collaboration with Sebastian's GLAD lab, we then introduced ELP6 mutations in vitro, and then we co-expressed these subunits in a cell-free system. And we found that this ELP6 mutation affects the expression and stability of the ELP4, 5, 6 subcomplex, as you can see here, where the mutation is present. But also the ELP6 mutation affects the stability of the ELP6 protein itself. And you can see here that there is a shift in the thermal stability of the protein. Then we isolated tissue from the mutant mice, brain tissue from the mutant mice and controls, and found that there are reduced tRNA modifications in the mutant mice these are specific elongated dependent tRNA modifications being reduced, whilst other tRNA modifications that are not dependent on this complex were not affected at all. So we did show that this specific ELP6 mutation perturbs the stability and the function of the complex. And then when we used electron microscopy to take a look at the degenerating neurons, we found the massive autophagy and these kind of electron dense material that is protein aggregate-like material. So we further performed immunofluorescence and found that indeed there is upregulation of molecular chaperones like protein 70 and ubiquitin. So there is some unfolded protein response in these neurons. We also found that ER stress-induced transcription factor CHOP is being overexpressed. Sorry, I just put it so I can't. Uh-oh. Sorry, I don't know. Okay, got it. Sorry about that. Let me, I hope you can still see me. So we also found that, yes, here it is, the CHOP is being upregulated and that this leads to apoptosis of these neurons as there is upregulation of cleaved caspase 3. So the mutation does lead to protein misfolding and aggregation, triggering UPR stress and Purkinje neuron apoptosis. Then just recently, we identified in collaboration with clinicians, we identified mutations in the ELP2 genes, and they're being responsible for motor defects, intellectual disability, autism, and epilepsy in these patients. And there is also global developmental delay. The mutations based on MRI imaging are causing microcephaly, which is reduced brain size, as you can see here, but they're also causing myelination defects. There is thin corpus callosum. We further modeled these mutations in mice and found that the mice have a very similar phenotype to this clinical phenotype, and there is a global developmental delay. There is a reduced body length and weight in the mice. They're just reduced in size. But then based on body to brain weight ratio, we find that these mice also have microcephaly. Then based on behavioral tests, we saw that there are severe motor defects in the mice. They also have limb clasping, but we were able to see repetitive behavior in the mice based on excessive self-grooming, which is very characteristic for autism. We analyzed their communication in ultrasonic frequency and found that there is a reduced number of calls. So they have communication deficit, which is again suggestive of autism in these mice. The mutations was quite severe at the end, leading to the lethality, being lethal in the mice. Then we employed diffusion tensor MRI imaging to analyze tractography in the mutant ELP2 mice, and found that we analyzed 5 major brain tracts and found that these cortical tracts were altered. Their organization was quite defective. But then we also analyzed the connection between different structures of the brain, and we were able to say that there is an overall hyperactivity in the brain connectome, which is again commonly found in autism. And especially there is a hyperconnectivity in certain brain regions like limbic system, which could explain aggressive behavior in the patients and self-injury. But on the other side, we have reduced activity or reduced connectivity in the brain regions that are responsible for social interaction, in some cortical regions also that are regulating motor function. Okay, so now we know what is the phenotype resulting from these ELP2 mutations, and that it also affects the brain connectivity, but that led us to further investigate potential neurodevelopmental defects as the mice have microcephaly. And indeed, we found that there is a reduced number of neuronal progenitor cells in the developing cortex of the mutant mice. And not only that these post-mitotic, which are DCX cells here, are being reduced in number, and some intermediate progenitors, but even the apical progenitors in the developing cortex that are giving rise to all these post-mitotic neurons are being reduced in number. So then when we analyze the proliferation status of these progenitor cells, we found based on BRDU and Ki67 staining that there is a premature cell cycle exit in the mutant mice. So they exit the cell cycle, but not only that, there's also an increase in apoptosis of these in neuronal progenitors, which then explains why we have less neuronal progenitor cells, eventually leading to microcephaly. In collaboration with Michael Piper's lab, we isolated 4 brains of developing 4 brains of the ELP2 mutants, and then we cultured them in vivo using a neurosphere assay, and we found that there is a reduced number of the neurospheres, and the neurospheres are also reduced in size, again confirming self-renewal defect in the mutant mice. Interestingly, when the neurons differentiate, there is also abnormal morphology of these neurons, as we found that their branching is being reduced by using by quantifying this. And we also found that the number of dendritic spines is being reduced, which would explain why there is a synaptogenesis defect as well, likely leading to all these phenotypic features we see in the mice. Then, just like in the wobbly mice, in the ELP2 mutant mice, we were also able to see reduced cerebellum in size. There is cerebellar atrophy due to a massive Purkinje neurodegeneration. And again, this is due to upregulation of UPR stress markers, CHOP ubiquitination, leading to apoptosis. Interestingly, when we analyzed myelin, because there is a myelin defect, not only in the patients, as I showed you on MRI images, but also the tractography analysis showed this aberrant organization of white matter tracts, pointing to, again, some myelin defects likely. We did see that there is reduction in myelin in the mutant mice. And we found that this is based on the immunofluorescence, that this is due to defects in mature oligodendrocytes rather than the progenitor cells. And the mechanism is quite similar to the Purkinje neurons. So there is UPR, we see protein misfolding and aggregation, eventually leading to apoptosis of these mature oligodendrocytes, which can account for myelin defects. When we studied these mutations in vitro, similar to the wobbly mice, mutations, we again saw that ELP2 mutations affect the stability of the protein, ELP2 protein, as there is again that shift in the melting temperature. But then we also saw in the brain tissue of the mutant mice that especially ELP1 and ELP2, which are subunits of the catalytic subcomplex, are being reduced in expression, whilst ELP4 is not that expressed. So it means it's not that being affected. So it means that predominantly ELP1 to 3 subcomplex is being affected by these mutations, as there is reduced expression of these subunits. Then we performed in vitro assay where we analyzed the affinity of the complex for binding tRNA, and acetyl coenzyme A hydrolysis, which is an important step in catalytic reaction where these moieties are being added to tRNAs. We found that when the mutations are present, there is a reduced affinity to tRNAs and reduced activity of the complex in terms of acetyl coenzyme A hydrolysis. And finally, when we analyzed the tRNA modifications using mass spec, again, similar to the wobbly mice, we saw that there is a reduced there is a reduction in these specific tRNA modifications that are dependent on the elongator complex, whilst the ones that are not dependent on it are not being affected. So this is a very elongator specific defect. And we performed proteomics analysis because elongator is a translational regulator, and we compared proteomes of the mutants and control animals. We found that there are upregulated proteins. Among upregulated proteins are those that are found to be linked to certain neurodegenerative diseases that are characterized by UPR, like Huntington disease, Parkinson's disease, whilst downregulated proteins have reduced cell cycle related proteins, also those proteins that are important for protein processes and DNA replication. So interestingly, among downregulated proteins, we also found that these are longer proteins. So longer transcripts are being specifically affected, and they're A codon enriched, which means that they're, again, highly dependent on the elongator complex. So what I showed you today is that elongator complex subunits and the mutations in these subunits lead to a wide spectrum of neurological diseases, including intellectual disability, autism, and epilepsy. That these mutations lead to neurodegeneration, myelin defects, microcephaly, and brain connectivity defects in mice. Likely the same things these defects can account for severe clinical phenotype in the patients. And that altered elongator function leads to reduced tRNA modifications, perturbing translation of long transcripts that are codon biased, so they're enriching A ending codons, triggering UPR and leading to impaired protein homeostasis. I would like to thank my lab, Wainwright Lab, and all the collaborators listed here. And thank you for your attention. I'm happy to take any questions.
Thank you, Marija. That was absolutely fantastic. So, Julian, over for you to questions, mate.
Thank you very much, Mike. Marija, thank you very much. I really loved that presentation. Beautiful data. What a broad and multidisciplinary approach. Looking at a very interesting mechanism that's clearly broadly impactful in neurodevelopmental phenotypic traits. And so the mouse seems an excellent model to investigate these molecular functions that could underlie behavioral phenotypes that you see in the human condition. So I'd like to open this session to questions. And so if I could ask Lisa, in fact, in the audience, to ask the first question, because she conveniently agreed with one of my questions that I posted beforehand. And then the next question will be from Massimo. Thank you. So first from Lisa, thanks.
Okay, so the question I had in there was about the wiring and the connectivity, which was really interesting in data. But why do you get the hyperconnectivity in the limbic system and then decreased wiring everywhere else? That's a really interesting phenotype.
Yes, yes, it is. Yeah, we were also at first surprised why there is such a massive hyperconnectivity, but then I looked up the literature, and apparently now DT-MRI is being used in autism quite a lot, as trying to kind of diagnose autism as early as possible. And there is hyperconnectivity in the patients as well. Now, I'm not quite sure how to explain exactly this massive hyperconnectivity, and especially in the limbic system. However, there are some sections, some parts of the limbic system being down with reduced connectivity as well. So it's not the whole limbic system being overactive, and down activity has been found in the cortex, also some parts of the cerebellum. So yeah, it's not just in the motor function regions as well. Sorry, I'm not sure if I answered your question. I actually really don't know why there is this massive hyperconnectivity, and I don't think that the literature is quite familiar with that as well. So this is just like a trend, something we see in autism, but we're not sure why this is the case. Yeah, it is interesting, definitely.
Yeah, really.
Thank you, Lisa, for your question. Can I go to Massimo, if I may? And Massimo, can you please ask your question, which is a very interesting one that I want to know the answer to as well.
Yeah,
Thank you.
Sorry, I'm just moving here so you guys see me. Can you hear me? Hello, everybody.
Yes, hi, Massimo.
Oh, I was just, Maria, I was just asking, so what do you think is causing the specificity of the LP6 mutation? Or is there specificity in the cerebellum, or is there, if you look at other tissue, and if there isn't, what is causing the specificity of that particular mutation?
Yes, that's a very good question. So, look, when it comes to the wobbly mice, the ELP6 mutants, we did look at other structures, and the mice do not seem to have microcephaly at all. So it looks like the hippocampus is also not affected. Somehow the cerebellum is mainly affected. Why Purkinje neurons specifically? At first we thought it has something to do with the codon bias, that the transcriptome of these neurons just uses specifically these codons that are A enriched, so they're dependent on the elongator. But then we found that granule neurons also have a very similar transcriptome in a way in terms of codon usage. So we are not sure. We believe that just Purkinje neurons, they're highly metabolically active. They're very sensitive to all sorts of stress, especially the protein misfolding stress. So there are other mouse models showing similar things, that UPR would just cause their degeneration, and those were germline mutations again. For some reason, they're very sensitive. I don't have an answer why exactly Purkinje neurons, but and yeah, interestingly, the ELP6 mutants didn't have defects everywhere else in the brain, really, but the ELP2 mutants do. This is likely because ELP123 is a catalytic subcomplex. So I think that the function of the complex is being more affected with the ELP2 mutations than ELP6 mutations. And we actually did show that now in vitro. So this is why the consequences are more severe in ELP2 mutants. And at the moment, we just believe this is all has something to do with the codon bias. As you could see in this proteomic data, that all down-regulated proteins are specifically enriched in A codons. So you see, it's a germline mutation, but in some cells, you will have higher codon bias for these, for A and codons than in other cells. So we think that's why also, oh yes, sorry, this is important. There was a nice paper published 2 years ago that showed that brain and the central and peripheral nervous system have really A and codon enrichment than other tissues for some reason. So that would explain probably why elongator mutations particularly affect these tissues.
Thank you.
Thank you, Massimo, for your question. Before I pass on to Tony for his question, I would like to encourage the students and the early career postdocs in the audience to have a question in hand and to have the ease within which to ask your questions. Please, we the ADNF have designed this program for you all to be able to summon your courage and ask your questions to any of these students. So please do that and think about it and as to have your question ready for after Tony has asked his. So Tony, can I please ask you to address Marija with your question? Thank you. It's a good one.
Thanks, Julian. Yeah, great talk, Marija. I was wondering with the vocalizations, I wasn't clear whether they were pup isolation or whether the vocalizations are still different in the adults and whether it's coupled to social interaction changes and whether any of it's sexually dimorphic.
Yeah, good question. So this was done in pups, just pups being separated from the mum. But we have not studied vocalization in adults. And this is because so we had these mutations on 2 different genetic backgrounds. On C57 mice, unfortunately, they die around P21. So that's why we had to use DBA2J, another genetic background, where they did live longer, but they're so severe motor defects that unfortunately that prevented us in studying not just vocalization, but also some tests like cognition tests or 3-chamber tests for social interaction. It gets really tricky because we did attempt these tests, but motor defects are so severe that we're not so confident whether the defects are there because of the phenotypes or the motor defects. Sorry, the motor defects is a phenotype or because of the mutations. So yeah, that was a bit tricky, but yeah, good question. We were wondering the same. Thank you.
Thanks.
Thank you very much, Tony. I will also indicate that the chat box is always open for anyone who has questions to any of the speakers during the session, which we can revisit at the end of the session when we have a few minutes after hand. So thank you very much for your questions, everyone. And Marija, thank you very much for your excellent presentation. Mike, I pass it on to you.
Thank you, Julian. And of course, thank you, Marija. That was absolutely spectacular. So going from one spectacular young scientist to another spectacular young scientist, no pressure, Sean, but you'll be okay, mate. We now have a change of gears moving to C. elegans research with Dr. Sean Coakley. So Sean is a Queensland boy, did his undergrad and his PhD at the University of Queensland, his PhD with Professor Massimo Hilliard, looking at elements of axonal regeneration and degeneration in C. elegans. Sean did very, very well during his PhD, and subsequent to that, landed himself a prestigious Dementia Research Development Fellowship to continue his research into this domain. He spent some time overseas recently in Kung Shen's lab at Stanford, again looking at different aspects of dendritic branching in this case, and neuronal development. And now he's back with Massimo, continuing on the amazing research they're doing, looking at different aspects of neuronal biology using C. elegans as a model system. Sean's title is Protecting Neurons from Mechanical Strain. And Sean, over to you.
Thank you, Mike, for the very kind introduction. Let me just share my screen with everybody. Okay, can everybody see that?
Yes, I can see it.
Yeah, perfect. So I'd like to start by thanking all of the organizers for giving me this fantastic opportunity to share my work with you today and providing this wonderful forum for us to interact in as well. So virtually every neuron, including those of the central nervous system, are susceptible to different types of mechanical strain insults. And mechanical strain itself has been implicated in the progression of several neurodegenerative diseases. Now, this concept in and of itself is particularly intuitive when we think about the peripheral nervous system, where neurons and their axons are subjected to quite obvious continuous mechanical stress due to their location within the skin, muscles, and movable joints. And this is very easy to think about if you think about me extending and flexing my arm. However, less intuitively, all neurons are actually experiencing mechanical strain, and this includes those of the central nervous system. But really little is known about the molecular mechanisms which protect against mechanical strain and ensure that neurons in the peripheral and central nervous systems are able to maintain their axons and the correct structure and function throughout life. So the work that I want to talk to you about today is work that we recently published earlier in the year, and I was very fortunate enough to have the opportunity to collaborate with 2 of my excellent colleagues in the Hilliard lab, Fiona Ritchie and Kate Galbraith, who I want to acknowledge here because they made crucial contributions to this story. So one of the major ways that neurons are inherently able to withstand any form of mechanical stress is proposed to be due to the membrane-associated cytoskeletal spectrum network. Beta spectrin forms part of this network, and together with alpha spectrin forms spectrin tetramers, which is shown here in magenta in this scheme. And in the axon, these are organized into a beautiful periodic ring-like structure that forms around the axon shaft itself in alternating rings with actin. Depicted here, actin is green in these alternating rings with spectrin in magenta. And this was first beautifully demonstrated with super resolution microscopy in hippocampal cultures from rat tissue, I believe. And since it's been observed to be very well conserved, not just across neuronal subtype, but also throughout the animal kingdom. Every species that's been looked at has this beautiful spectrin localization. So C. elegans are an excellent system in which to study how neurons resist strain. Their invariant cell-cell connections coupled with the beautiful single neuron resolution that we have allows us to really interrogate this in great detail. In C. elegans, specialized touch receptor neurons, such as the one depicted here in green, are subjected to mechanical strain due to the sinusoidal body movements that the animal makes as it moves. And this provides us with a very simple model in which to study the effects of this mechanical force from movement on neuronal development and maintenance. So all the work that I'm going to be discussing today is focusing on these so-called posterior lateral mechanosensory neurons, or PLM, which are a pair of bilateral sensory neurons that mediate the animal's response to gentle touch, in particular in the posterior half of the worm. The cell body of PLM is in the tail of the animal. It has this short posterior process and a long axon that extends into the midbody of the animal with this short ventral branch where it has synapses onto downstream neurons. This cell has a very complex and intimate relationship with its surrounding tissue, which happens to be epidermal tissue. It's a sensory neuron in the periphery. And if we take a cross-section through this PLM axon here, where this dotted line is, you can see that it actually sits within this epidermal tissue. And if we take a bit more of a close-up look at this left-hand side of the animal, this is now the head of the worm is into your screen and the tail is towards you. You can see this cross-section in green, the axon's actually embedded and ensheathed within this pocket of epidermis, which is known as the epidermal furrow. And this furrow runs the entire length of the PLM axon. This neuron's mechanosensitive, and as such, it's both physically and mechanically coupled to this tissue, which is important for both its development and its function. And it's coupled by these specialized attachment structures, which are analogous to vertebrate hemidesmosomes, which I'll talk about a little bit later in my talk. In the worm, beta spectrin is coded for by the gene UNC70, and elegant work from many labs have demonstrated that it functions to maintain axon integrity by mediating the neuron's inherent ability to withstand mechanical strain, primarily functioning cell autonomously within neurons. In spectrin mutants, this sensory neuron PLM is no longer able to maintain its integrity and undergoes spontaneous axonal breakage simply in response to the movement, the body movement of the animal as it crawls. So this is an example of two siblings carrying a semi-dominant mutation in UNC70, which affects tetramization with alpha spectrin. And we see a very low penetrance of axonal breaks in this animal. It's a very mild phenotype, but it's completely distinct from wild type. So in 5 to 7% of animals, we see this very low rate of axonal breakage. This is an example of these breaks. And this is a sibling with the same genetic background. And most of the animals look like this with a completely intact PLM neuron. So genetic screens aimed at isolating mutants with spontaneous axonal breakages like this really have yielded only a handful of genes. And this is true for all organisms, not just in the worm. So we've begun screening in sensitized backgrounds to overcome any potential redundancy. So I used this UNC70 mutant as a sensitized background and performed a forward genetic screen aimed at identifying mutants where this rate of axonal breakage was enhanced compared to this background. So from this screen, we identified a novel mutant, which we determined to be an allele of the Rab GTPase activating protein TBC10. And it presents with an absolutely massive enhancement of axonal breaks compared to UNC70 mutants alone. So this is about a 10-fold increase. We see now 70% of the worms have these axonal breakages in PLM, and these are the types of breaks that we see. This catastrophic break followed by degeneration of this distal fragment. So in addition to the allele that we isolated, this so-called VD31 allele that we isolated in Massimo's lab, we obtained 2 additional deletion alleles of TBC10 from the C. elegans knockout consortium, which further confirmed the causality of this TBC10 mutant, as they both present with significant enhancement in the rate of axonal breakage in the UNC70 mutant background. So what is TBC10? Well, TBC proteins are Rab-specific GTPase activating proteins, or Rab gaps. And in other systems, they've been demonstrated to regulate a raft of intracellular trafficking processes, such as microvillus structure, exosome secretion, and melanosome transport. And they regulate the activity of a target Rab by facilitating its conversion to its inactive GDP-bound form. So Rabs are active when they're in their GTP-bound form, and TBC10 facilitates the conversion to its GDP-bound inactive form. And this is the opposite reaction that the guanine exchange factors, or GEFs, participate in, where they activate the Rabs in the opposite way. Vertebrate homologs of TBC10 have been reported to function in lots of trafficking processes. As I mentioned, most recently published is exosome secretion, and more recent work in C. elegans has identified a role for TBC10 in phagocytosis, which is particularly interesting in the context of axonal degeneration. So we wanted to understand how TBC10 was functioning in this context of axonal maintenance and degeneration. Firstly, to test in which tissue TBC10 was functioning in to protect axons from spectrin-induced degeneration, we generated mosaic animals expressing a wild-type copy of TBC10 in different tissues. Firstly, we expressed TBC10 under its endogenous promoter, which provided a complete rescue of this axonal breakage phenotype. However, surprisingly, when we expressed it within the PLM neuron selectively, we saw no rescue of this axonal breakage, suggesting that TBC10 is not functioning cell autonomously to protect the neuron from damage. Curiously, instead, we see a full rescue of this phenotype when we express wild-type TBC10 within the surrounding skin, which is able to fully rescue these axonal breaks. And this was quite a surprising result, and strongly supports a model in which TBC10 functions in the surrounding epidermis to protect this embedded neuron from degeneration induced by spectrin dysfunction. Now, TBC10 shares a high degree of sequence homology with the human orthologs. TBC1D10A through to C is the human family. And you can see here, this is the catalytic region of this Rab gap. And shown in yellow is the conserved residue, which is mutated in the allele that we isolated. So we next asked if these human versions can also rescue the TBC10 defect when expressed heterologously in the worm. And of the three, two of them, A and B, are able to rescue this defect, while C doesn't, suggesting that in this context, the function of this family is conserved. So very briefly, I just want to touch on something important for this story. This TBC10 defect that we see of these enhanced axonal breakages is completely dependent upon spectrin dysfunction. We see no axonal breakages in single mutants of TBC10. However, surprisingly, spectrin in this context is also functioning non-cell autonomously, which is in contrast to how it's sort of understood to function to mediate strain. It's functioning in the surrounding skin as well, in complete synergy with TBC10. And I'm happy to take questions about this later, but for the purposes of this talk, everything I'm showing you is in a spectrin mutant. So after determining that TBC10 was functioning within the skin, we next asked where it localizes within this tissue to mediate this protective role. We did this using CRISPR genome engineering. We tagged the endogenous TBC10 with a GFP and a flag tag and visualized its localization by spinning disk confocal microscopy. So shown here is a lateral view of the tail of the worm. We have visualized the PLM neuron with a cytosolic tag RFP molecule, which is pseudocolored here in magenta. You can see the cell body here and the axon extending into the midbody of the worm. So we observe this tagged TBC10 at the borders of the epidermal tissue, particularly at the membrane of the epidermis. But I want to draw your attention to these regions that are highlighted by these white arrows, where it appears that the GFP is almost cascading off the axon, a bit like a waterfall. Now, this orientation in a lateral perspective, this is a large Z stack that's been visualized as a maximum projection, and it doesn't really give us much spatial context. So I'm going to flip this stack now, and we're going to look at it orthogonally. So now the head of the worm is into the screen, the tail is towards you, the lateral side is to the left, and the medial side of the worm, the center of the worm, is in the middle. And if we take a view of this region where the PLM axon is embedded within the skin, we actually see TBC10 localizing to what we think is the membrane of this epidermal furrow in which the PLM axon is embedded within. And you can see the axon here in cross-section in magenta. So this molecule is functioning within the epidermis to protect this axon from axonal breakage due to spectrin dysfunction, and localizes at the membrane of the skin in very close proximity to the axon. So we next asked what the Rab target of TBC10 was. Rab35 has been shown to be a specific target of TBC10 in vertebrate and invertebrates in a range of contexts. So in animals lacking TBC10, we expect that because this enzyme is inactivating Rab35, we would expect an increase in the pool of active Rab35, which in our context leads to an axonal breakage phenotype. So to test if Rab35 is a TBC10 target, we would expect that further loss of Rab35 would rescue the TBC10 UNC70 phenotype and prevent breakages. And this is exactly what we see. So compared to the TBC10 UNC70 mutants, animals carrying an additional null allele of Rab35 have a complete rescue of this axonal breakage phenotype. To confirm that Rab35 is also functioning within the skin, we expressed a wild-type cDNA of Rab35 cell-specifically in the epidermis in these triple mutants, and we were able to completely restore these axonal breaks back to the mutant levels, suggesting that it's also functioning in the epidermis. As we did with TBC10, Rab35 is highly conserved in humans, and the human version of this, the human cDNA, is also able to restore this phenotype back to the mutant levels when expressed in the skin. So TBC10 is functioning in synergy with UNC70 in the skin of the worm via this Rab GTPase, Rab35, to protect sensory neurons from damage. So to get at how it was doing this, we next asked where this effector Rab35 was localizing within the epidermis. To do this, we expressed a fluorophore-tagged version of Rab35 cell-specifically in the skin of the worm. We used this red fluorophore MK2, which is shown here in this image, pseudo-colored in magenta. We did this in a Rab35 null background to try and minimize overexpression defects. And what we see is Rab35 localizing to a mix of punctate and tubular structures throughout the epidermis. But again, if we focus on an orthogonal section and look at where this PLM axon is actually embedded within the skin, again, we see Rab35 localizing very similarly to TBC10 within the membrane of this epidermal furrow that wraps around the PLM axon. Also present in these images, which may be hard to see depending on the size of your screen, in the ventral and dorsal parts of the animal, this corresponds to regions where the epidermis and the muscle are attached to each other. We see a very faint but distinct circumferential localization of Rab35, which to us was reminiscent of specialized attachment structures that are in the skin that are analogous to vertebrate hemidesmosomes that I mentioned in my introduction. As well as mediating attachment of the skin to the muscle, they also are present along the length of the PLM axon and mediate its attachment and mechanical coupling to the epidermis. So this prompted us to visualize these structures and see if adhesion and attachment was potentially affected in our mutants. So again, back to this scheme of PLM, if we look at it within this epidermal furrow, these hemidesmosomes form these transepidermal attachment structures which link the skin to the neuron. And these are the proposed molecular components of these junctions on the epidermal side, as well as the axonal side that span this specialized extracellular matrix between them. And this has been very well studied in the worm, mainly in relation to where the skin and the muscle attach to one another in the ventral and dorsal aspects of the worm. These are the muscles here in this sort of bluey purpley shading. But less well understood is the linkage between the axon and the skin and its effect on development and neuronal maintenance. So the major organizers of these structures are the plectin homolog VAB10, which links intermediate filaments within the epidermis to this membrane receptor LED805, which is proposed to link across this extracellular matrix space with integrins on the axonal surface. So we obtained CRISPR engineered fluorophore labeled versions of both VAB10 and LED805 and visualized these in our mutants to observe if there was any effect on attachment. I'm just going to show you the VAB10A results today in the interest of time, but they were exactly the same as the results we saw with the tagged LET805 lines. So this is endogenous VAB10A labeled with GFP using CRISPR. And this has been demonstrated previously to localize in this periodic striped circumferential pattern in the ventral and dorsal aspects of the skin where it attaches to the muscle. And then in this very beautiful periodic punctate and stitch-like pattern along the entire length of the PLM axon. So this is how it looks in the L4 stage, which is the final larval stage before adulthood. And this is 1 day or 24 hours before we see the appearance of breaks in our mutants. So if we look in our mutants at the localization of this molecule, we can see a disruption to this attachment as we hypothesized. We can see a loss of this VAB10A, and we see the same thing with LET805 in discrete regions along the axon, and a disruption to the regularity of this pattern. If we look 1 day later in adult animals, these obviously all have completely intact neurons, and we now see a continuous localization of these attachment structures along the entire length of the axon. But in our mutants, we see a complete loss of attachment in intermittent regions. So there are portions where the axon's attached, but there are also these large regions where the axon becomes unattached. And in this particular example, the axon hasn't broken, but you can see that where it's not attached, it's starting to kink. And we think it's these regions of high strain as a consequence of being unattached that are causing the axon to break. So to summarize the molecular elements of this, these hemidesmosomes are known to be tension sensitive, and pathways have been identified in other tissues that can potentially modify the stability of these junctions in response to strain. However, the actual tension sensor in this system isn't known. So the idea here is that in response to tension after movement, these junctions may be stabilized or become more mechanically resilient in order to not rupture during movement. And I didn't have time to show you all the data today, but we determined that not only were TBC10 and RAB35 acting to regulate this hemidesmosome stability in response to strain, but spectrin was also functioning non-cell autonomously, very surprisingly, within the skin in synergy and in parallel with this TBC10 pathway, such that when both of them were affected, we saw a complete absence of these attachment sites after movement. So if we think about this at a more macro level, when attachment is regular and along the entire length of the axon, the worm is able to move normally and resist this strain. These junctions are mechanically resistant and nothing happens to the axon. But in our mutants, the movement induces a loss of attachment in these intermittent regions, which then we think the axon is experiencing regions of high strain and tension, which then leads to rupture and subsequent breakage and degeneration of this axon. So the ways in which non-neuronal tissues, glia in the central nervous system and the peripheral nervous system and epidermal tissue in the periphery as well, contribute to circuit assembly and maintenance in the nervous system are really only just beginning to be uncovered. So using these tools and this kind of model and expanding it within the C. elegans nervous system, I'm hoping to further uncover the molecular mechanisms by which this is achieved in my own lab. And I'm currently on the job market for independent positions in this very strange time to pursue these questions. So with that, I'd like to thank everyone in the Hilliard lab, especially Massimo for his incredible guidance, mentorship and sponsorship throughout my time with him, Fiona and Kate, who worked tirelessly on this project with me, and the rest of the group for the excellent environment they offer. We had help from all of these people technically for sequencing and microscopy, and our wonderful colleagues and collaborators around the world who provided strange and reagents. In particular, Kang Shen, as Mike said, I spent most of last year being hosted by him in his lab, and it was a wonderful experience. And I want to thank you all for your attention. I'll take any questions.
Thank you very much, Sean. That was really fantastic. I'm now going to open the floor for question time. And I can see, thank you for those that have put their questions on the chat box. I will start with asking Helen, if I may. Can you please ask your question to Sean and address him directly? Thank you.
Hi, Sean. Yes, that was a great talk. So in general epithelial biology, and it's also probably true for neurons, that coherins and their interaction with the catenins are a major modulator of tension between cells. So have you thought to have a look to see if there's any involvement for that system in your attachment between the axons and the epithelium?
Yeah, thank you, Helen. That's very true. And we haven't looked at cadherins in this context. At this particular, the epithelia is polarized as it is in many systems. And so I guess this junction is almost on the basal side, but it spans to the apical side. At the basal side of the epithelia, it's supposed to be very well characterized that these hemidesmosome components are mediating the majority of this attachment, and our data certainly supports that. We see a complete loss of attachment when these are disrupted. Cadherins have roles at other junctions within the apical side, I believe, and in other tissues. We haven't tested them directly, but I think especially if we try and move this model into other neuron glia pairs or other neuron epidermis pairs, we might want to look at these other attachment molecules to see if they're having a role.
Yeah, just to note, you do see some cadherin on the basal surface in polarized epithelium.
Yes.
So it's potentially there, yeah.
Yes.
Thanks.
Thank you, Helen, for your question. I